using synthetic biology approaches. Turn it over to you, Spencer.
Thanks, Bob. I think you said it all. I don't have to go into too much detail. So it's a bit of a long title, and I see a lot of the students who are here this morning in the lecture, so you're going to see a few of the slides which you saw this morning again pop up today, so sorry about that, but there is some new stuff as well. So a more simplified version of this title comes from my colleagues, where in relation to the Aboriginal Dreamtime is that Rubisco Dreamtime. You know, am I kidding myself in believing that we can actually improve Rubisco function? And hopefully by the end of the lecture, you'll be able to see strategies on how we're doing this. Well, in some respects, we've already done it, but and hopefully you'll feel, yes, there is a chance that we can actually improve Rubisco in plants, and in, you know, which may or may not help to improve productivity. So as Bob said, I'm part of this ARC Centre for Translational Photosynthesis. So I just thought I'd just explain what that is. It's the Australian government every 3 or 4 years funds nationally about 5 or 6 centres of excellence, where they invest heavily to promote collaboration between professors, you know, or chief investigators, or CIs as I've called it here, from all different universities around Australia. So within our centre, we actually just have 4 programs. The first one is improved CO2 fixation. So you can imagine this is the one which I'm heavily involved in. Increase light capture, and then exploit natural variation. So this is where we're sort of taking a mixture of using transgenic approaches and using natural variation, which we can have in a lot of our screening populations of sorghum and wheat, and also rice, which our industry partners are part of this. So CSIRO is the big Commonwealth Science National Institute in Australia, and IRI is the International Rice Consortium in the Philippines. And you can see that we have members from University of Queensland, Sydney, and up in, and 2 in Sydney. And our 4th thing is actually optimising the models, so that we can actually model what will happen if we make changes to the photosynthetic properties, how might it actually influence the canopy, you know, the crop, rather than just at the leaf photosynthesis level. So it's actually been really impressive. We're just now just over our halfway mark, and in every program there's been actually really great advances. So hopefully by the 2020 we'll actually have met many of our goals. So just because at the end I always run out of time, the work which I'm going to quickly talk about today involves a whole range of collaborators here, which are on the bottom. So these are, you know, a lot of international and some national ones. And this is my current team. Actually, I only have 1 postdoc now. Elena's just left me. Technicians and a team of really good PhD students who aren't actually doing any of the projects I'm talking about today. Some of the work is mainly the collaborators' work of Rosemary, Birch, and Brendan and Elena. So I just thought I'd set the scene as to why, you know, improving crop productivity is important. And this is this statement which came from Megan Clark, you know, almost a decade ago. So in the next 50 years, we'll have to produce more food than we've consumed in the history of humankind. So that just sort of sets the scene for the level of investment and improvements in productivity which the globe is going to have to deal with. And that increase is due to this increasing global population, which, you know, in theory, we're going to hit about 9 billion by 2050. So although the 60s, you know, that was great that we had the Green Revolution and suddenly we're able to outproduce our global needs for our dietary intakes of, in this case, you know, wheat and rice, the 2 main grains consumed, maybe not necessarily here in the US. But it just comes down to this statement here that global yield or progress at the moment is generally under 1% per annum increase. But to reach this target here, it needs to be up near 2 to 3%. So therefore, we actually need like a step change in the capacity of plant productivity. So a number of years ago, it was proposed that this can be done by improving photosynthesis, so it can help reach those targets. And there's various levels of evidence to suggest this is this is feasible. So again, if we just go back to this very simple equation, you know, photosynthesis involves the fixation of carbon dioxide, water to produce sugars and oxygen. So this is what supports life on Earth. And of course, one very important enzyme in this whole process is Rubisco. This is the gatekeeper between carbon dioxide, inorganic carbon, into starting the fixation of sugars, so production of organic carbon fixation. So today I'm just going to break my talk into 5 different sections. And again, I'm not assuming everyone knows all about Rubisco, even though it is the most important abundant enzyme on Earth. And then we'll just go through these other topics about how to identify amino acid changes which confer changes in the catalytic properties of Rubisco. How do we engineer Rubisco in plants? It's not necessarily a straightforward process, has various challenges. Point out that Rubisco needs to maintain its close relationships with a lot of the proteins it interacts with, and also just give you an example where we can demonstrate that, yes, it is feasible to improve the carboxylation properties of Rubisco by laboratory evolution, and maybe what will we do after that. So if we look at the fixation or carboxylation reaction of Rubisco, it involves the carboxylation, addition of CO2 to a 5-carbon substrate to produce 2 molecules of 3-phosphoglyceric acid, which feeds into the Calvin-Benson cycle. In this, following from this carboxylation phase, we have this reduction phase, which consumes a lot of energy, which is supplied through the light reactions of photosynthesis. And obviously, the other byproduct of these light reactions is oxygen. Once we've made these sugar precursors, these triphosphate, glyceraldehyde 3-phosphate, some of it goes back into remaking REBP, and whilst others it's then made to use the sugars which support the growth and productivity of a plant. So this process is often limited just by the rate at which Rubisco does this reaction. The reason for that is that Rubisco is actually a relatively slow enzyme. In plants, it only catalyzes around 2 to 5 cycles per second. And as a consequence, plants invest a lot of their resources into making enough of the enzyme to support adequate rates of photosynthesis. So that's why it's a big target. Another deficiency of Rubisco is that oxygen actually competes with carbon dioxide for fixation to REBP. And one of the products of the oxygenation reaction is 2-phosphoglycolate, which is actually toxic to plants. So therefore, the recycling of this process, of this compound back into 3-PGA occurs by the photorespiratory cycle, which actually traverses 2 other organelles. And during that process, though, you actually lose fixed carbon dioxide, and it actually consumes energy. So it's sort of resource inefficient. So ultimately, our aim, I should say, that we are trying to achieve is actually increase the flux of the carboxylation reaction by Rubisco and reduce flux through, or not necessarily eliminate, the oxygenation reaction. Then hopefully this will enable us to increase the flux through the Calvin cycle, either by its own or by actually tinkering with other enzymes within the Calvin cycle itself. So this is, as we can see, if you actually grow rice, this is just an experiment done at Erie, and the rice plants, I've put them on the side, they don't grow, vertically. At increasing CO2 concentrations, we can see that plant growth continues to increase. And that's because of this factor here, that we actually increase the flux through the carboxylation cycle of Rubisco and reduce the oxygen side of the cycle. So therefore, we actually reduce photorespiration, and the associated energy costs means that we're saturating the carboxylation kinetics of Rubisco. So this is sort of an indication that if we can actually do this, that we should actually see improvements in productivity. Other ways which you can do it is actually just simply concentrate CO2, more CO2 around Rubisco and the chloroplast. Now, some plants have done that, and some organisms, so I don't know if you know about C4 photosynthesis, but your maize, your sugarcane, they actually have this capacity. They sequester Rubisco in a special type of cell, and they have a process using another carboxylase enzyme to produce acids which decarboxylate and produce higher levels of CO2 around Rubisco. Of course, what that means is these plants usually have higher turnover rates and there's a lot lower rates of photorespiration. Similar systems are in algae where they have pyronoids, which have a similar CO2 concentrating. They localise Rubisco and carbonic anhydrase inside these structures. And also in carboxysomes in cyanobacteria. Again, there's special protein shells where Rubisco and carbonic anhydrase are localised in an area so that you can elevate the CO2 levels around Rubisco like 20 to 40-fold that within the atmosphere. So the big question is, how do we go about, building a better Rubisco? And this is something which has consumed essentially my whole academic research career. And along the way, it's been really fascinating. If I knew now what I knew, if I knew then what I knew now, I'd be a lot more excited than what I was back then. Because the technology, the advances, synthetic biology has provided us with so many new tools and capabilities. So essentially, our aim is to just target two main parameters, really three, is to improve the carboxylation efficiency. So that's the speed of the enzyme divided by its substrate affinity, and also the specificity. So essentially, these are the parameters which we need to enhance. So that in plants like wheat and rice, who have a C3 physiology, that is, they don't concentrate CO2 around Rubisco, we should be able to improve their photosynthetic properties. Now, of course, we know that there are different forms, the structural forms, and also in terms of the catalytic properties, but they vary in nature. The form which we're most interested in is this very large Rubisco molecule made up of 8 large subunits, shown here in green, and 8 small subunits. So you have tetrads of smalls at the top and the bottom, and they're required for stability and actual catalytic function. So they're found in plants, algae, cyanobacteria. Then we have these other forms, so these form 2 Rubiscos, which don't contain small subunits. They also play a photosynthetic role, and they're generally found in photosynthetic bacteria, which live under very low oxygen environments, as well as in dinoflagellates. So this is the one which I discovered during my PhD. And then in the archaea lineage, and as you can see here, it's believed that all Rubiscos evolved from an archaeal origin, but this is a non-photosynthetic role. These organisms produce Rubisco to soak up REBP that's produced during other metabolic processes, such as nucleotide metabolism. And so these have actually evolved quite distinct kinetics from here, because their focus here is to actually increase affinity for REBP, and these ones here are trying to fix more carbon dioxide. But what is shared amongst all those different forms is that catalytic chemistry. So again, we have these competing reactions, CO2 binding to the enediol form of REBP once it's bound to the active site, goes through a series of 5 partial reactions to produce our 3 PGA molecules, which go into photosynthesis. Or if oxygen competes successfully to bind, to interact with the C2 position on the REBP, the enediol form, then we get PGA, 3 PGA and 2 phosphoglycolate. And as I said before, so one of the rationales which we put forward many years ago is that maybe one of the impeding factors for Rubisco's inability to keep up with the changing CO2 and O2 pressures in the atmosphere is maybe because of this complex catalytic chemistry. As you can see here, the enzyme does not actually bind either of the gaseous substrate. It only actually binds the REBP. And the whole focus is to bind the REBP and orientate it in such a way that it can get attacked preferentially by CO2, which is less abundant in the atmosphere, about 400 ppm, over O2, which is around about 20,000 ppm. And of course, there's this extra cost here because of this photorespiratory mechanism that we actually get loss of fixed CO2. So as I mentioned in the lecture this morning, in plants like wheat, around about 30% of the CO2 that's fixed during photosynthesis is actually lost through photorespiration. So the reason why they have this conserved catalytic mechanism is that they all have a single origin. As I said, it's probably an archaea type Rubisco, which evolved around about 3.5 billion years ago. And of course, back then, the Earth's atmosphere was very high in CO2. Then, of course, oxygenic photosynthesis evolved, and so suddenly we had oxygen being produced in the atmosphere, and unfortunately oxygen is a competitive inhibitor of CO2. So Rubisco's had to evolve under these very contrasting, changing levels of CO2 and O2 in the atmosphere, and hasn't been able to, one might presume, keep up very well. So the reason, a consequence of having this same catalytic chemistry is that if you actually look at the catalytic site in all those different types of Rubiscos, although they have quite varying CO2 affinities, turnover rates, their active sites are very highly superimposable. That is, they look very much the same when you look at their crystal structure. And so this is what has actually stymied a lot of the Rubisco research over many years, is because it gave no clues as to what causes this catalytic variation. And if you actually look at within the large subunit and you look at the location of these residues here within the large subunit, you can see that they're actually interdispersed all over the large subunit. So here we have what they call the N domain and the C domain of Rubisco. It's about 150 and about 300 amino acids. And this is where you find the catalytic pocket in this subunit here. Whereas here, the small subunit, remember the ones which are at the top and the bottom, none of their residues actually contribute to the active site. But as I said before, it's critical that the small subunits to bind to actually enable activation or catalytic activity within the large subunit sites. But that's not the case in the form 2 and the form 3 enzymes. So how do you go about identifying catalytic switches then? If we look at the crystal structure and we can't really get much out of that, then what you need to do is you need to survey. You need to survey both sequence and also catalytic function. You can't just determine these just by looking at sequences, or you can't do it very effectively. So essentially our pipeline of research is survey for natural variability. So we survey plants, we survey algae, we survey bacteria. And then by making correlations between what kinetic features correlate with what amino acid changes, we can start to identify potential solutions that might be responsible for the different catalytic properties. And then, of course, we need a way which we can actually test this in a planter. And so that's pretty much, as Bob said in the introduction, is a technology which I brought to the ANU a number of years ago. So if we actually look at the exploration thing, there was a bit of a flurry. They used to call it stamp collecting. Why am I going to fund someone to go out there and screen kinetics of Rubisco and get their sequence? So it was very hard to actually get any funding for that. So you always sold it in a different way. So again, we've done a lot of screening of different algal Rubiscos, as well as there's been a number of papers from groups over in Europe where have looked at the kinetics of different plant Rubiscos. So one of the reasons why we have high hopes that we can actually improve plant Rubisco is that there are better forms out there in nature. So for example, here we can see in some red algae, these filamentous red algae, this one here, Griffithia monolus, this is another Coelacanthus and another Borastichia, and they all have very good kinetics. And their kinetics would suggest that within the context of a leaf, where as you increase the CO2 concentration in the leaf and you see the photosynthetic rate, these Rubiscos would actually outperform the plant enzyme, okay? And by a substantial amount. The word of caution here is that when we analyse these diatom Rubiscos, and this is a paper published a couple of years ago, is that these diatom Rubiscos actually have a higher specificity than plant Rubisco. And everyone thought, oh, they're going to do better enzymes. But they actually have really poor carboxylation efficiency. So therefore, although they're quite fast enzymes, they have very low affinities for CO2. So using models which we can simulate photosynthesis in plants like wheat and rice and tobacco, we can predict that Rubisco is not going to be any good within the context of a plant. When you actually survey the properties of some plant enzymes, you can actually find some examples where, hey, in actual fact, its catalytic properties will outdo a C3 organism. So since tobacco is our model organism, or model test system, and that'll become evident soon why, when we actually measure the kinetics of Rubisco, such as those from maize, we can actually find out that, hey, if we actually put a corn, Rubisco from corn, into a tobacco plant, in theory, its photosynthetic rates would be higher. But it's not the case for all faster enzymes. So again, this is another enzyme from another C4 plant, which has a high turnover rate, but again, it has a low affinity for CO2. So it just means that its photosynthetic rate is around about the same as tobacco. So we use these sort of steps where we measure the kinetics, we look for amino acids, we use these models, so we simulate, and then we start to say, well, that one's interesting, that one's not, and we go from there. But we also, everything we do at 25 degrees. That's a big challenge in the literature. So these are sort of like the kinetic properties for tobacco Rubisco over a different temperature range, so between 0 and 37 degrees Celsius. And you can see that K-cat goes up with temperature. The affinity for CO2 reduces with temperature, that is, the Kc goes up. And we see that the carboxylation efficiency, increases with increasing temperature. It starts to hit a plateau above 25 degrees. And the selectivity for CO2 over O2 actually decreases with temperature. So therefore, it becomes more of an oxygenase enzyme the higher the temperature. Now, if we actually, what we did is we looked at a whole range of, there's about 15 different grass species from Panacea. These are Australian grass species. But what they have is they have different physiologies. They have C3, C4 intermediate physiology, or they have representatives of each of the C4 types of photosynthetic physiology. And all I want you to take home is that you can actually see that there's a dramatic difference in their catalytic properties and how they vary with temperature. And if you actually then go and take all these kinetics and you put it into the photosynthetic models, you can find some examples such as different types of plants from the NADPME lineage of C4 plants, which may provide a benefit to C3 plants at high temperatures. So how do we then, if we find these potential solutions, how do we actually go and test them, get a proof of concept? So we do that by a process called chloroplast transformation. So if we actually look at the process of Rubisco assembly in plants and sort of simplify it, we see the large subunit, this is the one which contains the catalytic site, the catalytic pocket, is actually encoded in the chloroplast genome, whereas the small subunits are coded within the nucleus. Okay, so this is generally in multi-gene families, usually 2 or more. Wheat has 37 copies because it's a hexaploid, tobacco 13. And so the large subunits are made inside the stroma, and the small subunits are made in the cytosol, transported into the chloroplast, and they're where they assemble into this hexadecameric enzyme of 8 large and 8 small subunits. So the most traditional way of doing any sort of structure function analysis is to express proteins in E. coli and do mutagenic analysis. Unfortunately, when you take the large subunit, shown here, and the small subunit, you put in an operon and express it in E. coli, is you won't get any assembly. You get the subunits made, but they won't fold and assemble into a functional enzyme. And this has sort of halted a lot of, or stymied or prevented a lot of research into engineering Rubisco, except for a few forms which can actually express an E. coli. And this is because, as we know, the folding and assembly machinery of E. coli, can't actually assemble the high plant Rubisco. And that's if we actually now look more in depth about what is actually involved in the folding and assembly of plant Rubisco. Actually, I'm not going to go into that yet. So most of the engineering we do, therefore, we have to do within the plant itself. And so we do that by this chloroplast transformation, which is the synthetic biology tool, which we have used for over a decade now with quite success. So chloroplast transformation is possible in a number of crop species: carrots, lettuce, soybean, tomatoes, potato. But the most efficient system is actually in tobacco. So that's why it's our model plant. We can generate tobacco from a single cell, and the efficiency of the transformation process is a lot higher than in these other species, and also a lot faster. Unfortunately, our target plants, which we're mainly interested in, the grain plants, particularly rice and wheat, chloroplast transformation through to producing homoplasmic plants is as yet untenable. But this process is not quick, and it's actually quite an expensive process, from doing the transformation of the DNA using the ballistic gun into the tobacco, through the screening process, to producing the plants, which we can then both analyse the Rubisco kinetics, as well as correlate that with the photosynthetic properties and the growth of the plant. So generally, we can already predict what this is going to be if we know what the kinetics and the content of the Rubisco are. And that's using the models to measure photosynthesis, which I'm not going to talk about. I talked about it this morning, so if you want to find out more, talk to one of the students at my lecture this morning, because I'm sure they all remember that. So a couple of the examples which we've gone to test the catalytic switches is a paper which was published a number of years ago, where we focused on a genus of plants which coded either methionine 309 or isoleucine 309 at a particular residue. And what we showed is that these just interchanging between these two amino acids actually conferred the difference in the kinetic properties between a fast and a slow Rubisco. The challenge here is that that is correct for a number of plant genera, but it's actually not across the board. It's not universal. So therefore, when we're looking at trying to find solutions to improve Rubisco catalysis, generally we have to be focused on our target of choice. So whether it be wheat Rubisco or, in our case, tobacco Rubisco. So that adds to the challenge, okay? There's another challenge in that what we also did, and this work was actually done as part of the Wright program, which I was part of, as subcontracted through Illinois, where we actually took the large subunit of a whole range of crop species, and we thought, well, let's just see, can we actually use tobacco as a surrogate? We can't transform in many of these species, or we can, but it's very, very slow. And so we'll do all our testing in tobacco. So this is just one of the first tests we did, and what we found is that in the plants producing all the different crop Rubiscos is they all grew at various rates. Some grew, wild type like, which is like potato here, and surprisingly strawberry also grew very fast. And the reason for that turned out to be that if we actually look at the phylogeny of the large subunit from all these different crops, and we sort of rank it relative to tobacco, we see that the further away we go from tobacco in terms of sequence similarity, we also see increasing reduction in the amount of large subunits that can fold and assemble to make a functional Rubisco. So again, this indicated to us that there's some, the folding and assembly requirements of Rubisco are fairly specific. And as I said before, unfortunately, all the monocot Rubiscos can't assemble in tobacco chloroplasts. And I'll give you the answer. To actually get a monocot Rubisco to assemble in a tobacco chloroplast, you need to make 6 amino acid changes. So one of my students did that for his PhD. Still not published. So again, in addition to this inability for the folding and assembly requirements of monocot Rubisco to be met in tobacco chloroplasts, when we also tested by introducing all these Rubisco operons from these algae and diatoms in tobacco, what we saw is that we can still actually get the subunits made, but they can't fold and assemble. So again, the folding and assembly requirements of even these algal Rubisco are sufficiently divergent from that from high plant chloroplasts. So that adds to a bit of frustration. But what it did show us that, and we realised this a number of years ago, that for us to continue on with our Rubisco, recombinant Rubisco expression studies, we needed to understand the chaperone requirements of Rubisco. Unfortunately for us, people like David Stern at Cornell and Manojit Hartel at Max Planck Institute in Germany had actually started to do this in earnest. Which is why Manojit Hartel's just got the Kettering Award from ASBMB. So here we have a biochemist, knows nothing about plant physiology, and she's getting one of the prestigious plant physiology awards because she's just made such a huge impact on the Rubisco field in a very short time. So what are these complementarity needs of Rubisco? So if we actually look at this assembly process of Rubisco in plants, it's actually more complicated than what I made out earlier. So the large subunit needs is translated in the stroma, undergoes a number of post-translational modifications, but also needs various chaperones to maintain it in an unfolded state to be delivered to the folding cage of the chaperone complexes. And then when they exit here, this is what's been a bit of a black box for a number of years. And it's only since 2007 onwards that we've made significant advances and found out that there are actually 4 chaperones which is specifically required for folding plant Rubisco. One of those, which is called Rubisco accumulation factor 1, when we looked at its phylogenetic tree and compared it to Rubisco large subunit, we found that they're almost a mirror image of each other. So this made us think that, hey, hold on, this sort of suggests that they've evolved in parallel. Okay, so that sort of indicates that there's some complementarity requirement to be maintained. So what we did is we noticed that the Arabidopsis RAF1 versions were quite different to that in tobacco, and at the amino acid level they're only about 60% homology. So we thought, well, let's test it. Let's use our chloroplast transformation capacity to look at this. So what we did is we generated a tobacco line where we replaced the RBCL gene from tobacco with that from Arabidopsis. As well, we made another line where we put in the large subunit, but we also put in its complementary RAF1. And then what we did is we compared how well these plants did. And if you actually look down here, so this is just the wild type control, which grew faster than all of them. Those plants which were just producing the Arabidopsis large without its RAF1 grew a lot slower than those producing both the large and the complementary RAF1. And this is just a growth curve here, to change in height over time. And this sort of correlated with the difference in their photosynthetic rate. Those which had both the large and its complementary RAF1 actually had higher photosynthetic rates than those just producing the Rubisco comprising the Arabidopsis large. And the reason turned out to be simply because these plants here were making more of the Rubisco, okay? This hybrid Rubisco comprising Arabidopsis larges and tobacco smalls. So understandably then, that's why they grew faster and had higher photosynthetic rates. So this is good indication or good demonstration that you needed this complementarity to be maintained to actually improve the assembly capacity of the large subunits. And then a paper came out again from Managit's group soon after that to actually resolve the crystal structure of RAF1 and show that it is actually interacts with the larges, and we're about to have a look at that now. So the question which we raised this in a review paper back in 2007, 2008, that we suggested that maybe the need for chaperone complementarity has also impeded or slowed the potential of Rubisco to evolve during evolution, or evolve during evolution. That makes sense. And so the work which has come out of the Hayahata lab has actually used, cryo-EM and crystallography to actually show the interaction of these chaperones with the Rubisco. So as we show up at the top here, this is what the final product looks like. We have our core of 8 large subunits, with 2 catalytic sites per dimer, and then we've got the small subunits arranged at the top. So what we have is this other molecular chaperone, acts like a molecular staple, comes in a dimer. It's a very small protein, only about 15 kilodaltons, called RBCX. And we showed many years ago that this actually stimulated the assembly of Rubisco, but we didn't know how. And what they showed is that it actually binds to the C-terminus of your nascent large subunits, and they staple the dimer of larges, which arrange head to tail, and then enable the formation of octameric core with another 16 BSD2 subunits around there. So you can actually see here that this actually provides great room for the small subunits to come in and bind at the top, and then, in theory, displace their bound RBCX. And that was the working model back in 2007 through to 2012. Likewise, there's another protein, and this one was only discovered last year. Well, it was discovered back in 1999, but its structural role was only published last year. And you can see here that it also acts as a monomer, but it also binds to the dimer, large subunit dimers, so that within an L8 core you have 8 BSD2 molecules bound. And it's also thought to act as a stabilizing factor, so that when small subunit binds, it also displaces the BSD2. What we can't explain is that the binding of BSD2 actually binds in regions which also RBCX binds. So there's a bit of an ongoing discussion about that. Then this Rubisco accumulation factor 1, initially discovered again at David Stern's lab, but then structurally characterized and mechanistically characterized in the Heyer-Hartel lab. You can see this forms a dimer. It's actually quite a large dimer. It sort of acts like a bit of a coat hanger from the top to the bottom of each dimer. And again, you can see that the binding of the small subunits is, has the potential to displace these bound RAF1s. And okay, so this is the protein which we showed you needed sequence complementarity to improve the production or biogenesis of Rubisco. So ultimately, this is what it's thought. The small subunit comes along and displaces these molecular chaperones, or RAF1 comes along, displaces these molecular chaperones, and then the small subunit comes, and I think I've summarised that in a second. But anyway, this sort of goes to show that Rubisco is actually highly dependable on having compatibility with its molecular folding partners for function. And the proof of the pudding for this actually was just published again last year in the same paper, citing the BSD2 structure, where they took the genes coding each of the folding partners, as well as the large and the small subunit from Arabidopsis. Again, as I said earlier, you can't fold Arabidopsis, Rubisco, and E.coli, but you can when you actually have all these other partners. So this is a pretty amazing discovery, and it got many of us very excited, some of us a bit upset, because we have also been doing this with tobacco and wheat Rubisco. So we have pretty good, well, we knew that it was going to work then. But it's not as easy as it looks. I'll give you that warning. They make it look very easy in the paper, but it's not. There's a few tricks. So yeah, where our focus is on wheat. So this is a paper which is just about to come out in biochemistry. Again, another review, but I think it's probably the nicest of the reviews, showing the Rubisco large subunit synthesis, the folding, and then interactions with RAF1, and they say possibly RBCX. RAF2 is another Rubisco accumulation factor. This is the one where we still don't know what it does, and we have some hints, but no one knows, or someone does, and they just haven't published it yet. And then BSD2, binds in theory and displaces, or in this case, RAF1 or RBCX. Again, this is all, conjecture, and then the small subunits come along to finalise the final product. So this is all very nice, and they showed in that E.coli work that if you just left out any one of these enzymes, these chaperones, you didn't get Rubisco produced, except RBCX. You could actually leave that out, but the yield of Rubisco was actually a lot lower than if you had it in there. So last thing I want to talk about in the last 10 minutes is improving Rubisco by laboratory evolution. I want to sort of tie it in with what we're doing. So a number of years ago, I had a PhD student, Oliver Muller-Kaya, who designed an E. coli strain, which is survival is dependent on Rubisco. So if we actually then think about directed evolution, I don't know if you know about protein-directed evolution, where you take a gene, you make a mutagenic library, you introduce random mutations, you transform that into your host. Generally, it's an E. coli strain, can be yeast, and then you actually assay for activity. And this is the critical part of this whole process, where you've got to actually have a screen which allows for high enough throughput for you to detect whatever you're looking for. Because unfortunately, the classic for directed evolution is you always get what you screen for. So you may not actually get what you're looking for, but you find out what your screen is actually screening for. So here I'm going to answer the question, can we do it? Yes, we can. So using the system I'm about to describe, is that we've actually been able to improve the kinetics of both Archaea Rubisco and Cyanobacteria Rubisco. So let's talk more about success. Yep. let's talk about the system. So the original systems which have been used, and a number of groups have used this around, there's one in Georgia, there's a couple in China, and what it does is it involves hacking into the metabolic pathway of E. coli, because you get the production of ribulose 5-phosphate through the pentose phosphate pathway. So if you introduce the enzyme phosphoribulose kinase, it actually catalyzes the formation of ribulose 1,5-bisphosphate. Now that's actually toxic to E. coli. How? We don't know. We don't believe it's because you're drawing away these resources. But as you notice, RUBP here is actually the substrate for Rubisco. So if you add Rubisco to the system, you alleviate that toxicity because it produces 3-PGA, which then feeds into pyruvate and normal cell growth. So this is the phenotype. Cells, add in PRK, nothing grows, add in Rubisco on top of the PRK, and we get restore growth. So we set this system up such that we can regulate the amount of PRK being produced by putting it under an arabinose-inducible BAD promoter, and then we regulate the amount of Rubisco produced using a LAC promoter, IPTG-inducible promoter. So if we do this and we have a selection system, then a better Rubisco would look something like this. The cells will grow faster. That's how you identify it. Or you do under conditions where the wild type won't grow and only those with better Rubisco activities will grow. And that's the beauty of this life and death system. Problem is, though, that in all the literature out there, which have just used this very simple method, is that it's like looking for a needle in a haystack, because you get transposon silencing of PRK activity is prolific. So you get a false positive frequency of about 0.5%. Now you may think that's not very high, but if you're plating out 10,000 cells, that's 50 colonies. If you're plating out 100,000 cells, then you get 500 colonies and you may get 1 positive and 500 negative. So to go search for that is quite a nightmare. And most other groups have fought through and gone through that nightmare. So what we wanted is we wanted a system where we got no false positives. So to do that, all we did is we just tethered antibiotic selectable marker onto the PRK. So it produces a fusion peptide, doesn't affect PRK activity, and codes for kanamycin resistance. We tried a whole range, but kanamycin turned out to be the best. So if you got transposon inactivation of PRK, then suddenly you no longer had kanamycin resistance, so the cells wouldn't grow if you have kanamycin on the media. So the simple experimental system, so that our target enzyme, which we tried first, was this Rubisco from Thermosynechococcus elongatus. We chose this one because it's probably got the best kinetics of all cyanobacteria Rubiscos. So what we did is we put the genes coding the large and small into this E. coli, and we had the PRK, kanamycin or MPT, neomycin phosphotransferase fusion being produced. And then we alter the amount of arabinose in the media such that we increase or decrease the amount of PRK and the RUBP toxicity level. And then we can also increase and decrease the amount of Rubisco by doing that. So anyway, under a standard set of conditions where we have LB with the antibiotics, IPTG, arabinose, this is a high RUBP producing concentration, grown high CO2. We screened around about 250,000 mutants on a plate, and we actually got in this experiment 15 colonies. And then we take those 15 colonies and we extract back out the large and the small subunit plasmid. We retransform it back in to make sure that whatever is conferring the faster growth, that it's actually due to that Rubisco. We sequence it. And what we found from those 15 colonies is that we got a number of mutants. Many of them we got multiple times independently. So this one here, this is a small subunit mutation here, here, here. And this is a large subunit mutation down here. So this is just when you take these cells and then you just simply grow 100 cells on these different conditions and compare it to the wild type. Nothing grows on the wild type. So that's making the wild type enzyme. So you're pretty confident that you've got higher Rubisco activity in these guys. Now that could actually come by increasing Rubisco levels in the cells or improved kinetics. So you go and you measure the Rubisco content, how much of the Rubisco is made in these cells. And we can see here that this mutant here, which had that valine 98 mutation, actually increased Rubisco production. The others not so much. But then what we saw is that the K-cat, or the CO2 fixing speed of all the mutants, was around about 10 to 25% faster than the parental enzyme. The carboxylation efficiency was again similarly improved. And in 2 cases, we actually improved the specificity for CO2 over O2. So this is actually the first example of a L8S8 enzyme with improved kinetics. There was another one published by the Hartog group, and we actually showed in this paper that they got it wrong. And that's because measuring Rubisco content is actually critical for this process. And she just had a very bad PhD student who got it wrong. Anyway, won't go there. So the amazing thing is, when we actually looked at where are all those amino acids located, we found that in the final structure, they're all localized in one area here. So this is the small subunit here in this, I suppose it's green color here. And this is one of the large subunits. This is another large subunit. This is part of the 2 largest. So the positioning of these amino acids is actually just shown here in red. So they actually all co-localized to an interface at the large subunit, small subunit, or at the large subunit, small subunit interface. So this is on the surface of the molecule. So the active site, oh, actually, there it is, here. So they're actually quite distant from the active site. They're not, abutting and close by. It's more than 20 angstroms at a minimum. So this is quite interesting because none of the other mutants which we'd ever done or in the literature had ever targeted residues in this region. Now you can say, well, have you gone and looked at the same region in plants to see, are these residues conserved and can we actually just introduce them into the plant Rubisco? And the answer is no. So again, there's no silver bullet. So unfortunately, although they form an L8S8 complex, the amino acid conservation around these areas is not very strong. So what do we do now? So up until now, what I've talked about is, we survey our kinetic, natural diversity in kinetics, and then we try and introduce those changes in. I've just talked about the large subunit. We also engineer the small subunit by various means, both chloroplast transformation and nuclear transformation, so that hopefully we can improve photosynthesis. But now the question is, well, now that we know previously we weren't able to assemble plant Rubisco in E. coli, well, what if we now, that's 2007, what if we now take all these, what we know here about plant Rubisco assembly factors, and we take all the genes, like they've done previously for in E. coli, and transfer them into this system, into this directed evolution screen system, so that hopefully altogether, this is a bit of a nightmare, engineering nightmare, but we're getting there, that we can actually start to engineer or do directed evolution on plant Rubisco, not cyanobacteria Rubisco. Identify the solutions and then use the tools which we already have to actually introduce it into the plants. Supercharging photosynthesis. So, in summary, yep, on time, cool. So if we go back to the title of the talk, surmounting the constraints. Yeah, it sounded good at the time. Now it doesn't sound too good. But anyway, on Rubisco evolution. So what we're trying to say is that maybe the evolution of Rubisco has been slowed by the fact it's got a very complex catalytic chemistry, but also the requirement for chaperone complementarity. So in some directed evolution experiments done within the Hartog lab, if they actually co-express one of those chaperones and put it through a system, the mutants which they select at the other end are totally different. Okay, so therefore, evolution is impacted by its molecular partners, the interactions requirement there. So our solution is, as I've just sort of tried to summarise on that last slide, is can we now use these 2 synthetic biology tools, the capacity to assemble plant Rubisco and E. coli, and our new directed evolution, tools to actually identify solutions for improving plant-based Rubiscos. And then we already have established methods for introducing these into various crops. So For plastome transformation, we do that in a number of different tobacco species, but also we've just started up establishing it in canola. And, you know, obviously nuclear gene transformation, just general transformation, as well as using CRISPR-Cas9 gene editing. We can do that in rice. Our colleagues across the road do it in wheat for us, and we're just also setting up doing it in canola as well. So it's a really strong tool. It's not necessarily the answer always, because if we want to make particular amino acid changes in the small subunit, you are still restricted somewhat on your guide RNA design. But we're also using this system to actually hyper-express small subunits, because you need about 8% of your protein has to be small subunits. So most transformations don't give you that. Thank you. Any questions?
How important is it that you express responsive
So we have various tobacco genotypes where we've knocked out small subunit production and transferred it to the chloroplast. At the moment, we can only get around about 60% wild type levels of Rubisco produced. And yeah, but for what we're doing, that's enough. But ultimately, we actually want to do it by nuclear transformation, primarily because nuclear transformation is much more amenable in crop species than chloroplast transformation. But it does provide us a nice tool. A lot of the work out there where they've already engineered or changed the small subunit by putting in a foreign one, they've still got their native population of smalls, and that can actually be, that complicates your interpretation about how amino acid change influences catalysis. So we came at it a different way, eliminated the endogenous smalls, and so we can focus. And the reason why we do it by chloroplast transformation, not nuclear transformation, is because we do it in combination with the larger. So we do the large and the small at the same time, so we can actually do different combinations all the time. This is work which, embarrassingly, we started when Lynette was working with us. So that's how long ago, and we still haven't published it. So we've gone, yeah, it's going really well. Peter.
I wonder how the shutdowns work. You have eight the other or the line. Just when you push that structure, I saw that all of that long.
Yeah, well, that's exactly right. So that second line, well, one of the latest slides, which is a review, a mini review coming out from one of my old students and manager at Hartle. They're trying to, based on what they've seen in the E.coli assembly experiment, sort of say, well, this could be the binding order of them. Personally, I don't think RBCX plays a role in the plant. We can knock it out. But when we add in a foreign RBCX, we can actually see it interact with Rubisco. So it's yeah. But it's expressed at such low amounts that biologically, I don't think it can have a role. Whereas BSD2, we know it does have a role, because we can trap complexes from the plant which have BSD2 down. So we know that that is a real intermediate reaction intermediate or structure intermediate. So they're the ones which usually occur pre-folding cage. So what they've shown in this work in E. coli is that the bacterial, KJE type complexes are compatible with the chaperoning function which they do. So they essentially bind to the nascent peptide and prevent it from misfolding. So they keep it in an unfolded state such that then they can just, shove it, not shove it into, give it to the chaperone folding cage. And so, yeah. Does that answer the question? Otherwise, I'll go on about other stuff.
This follow-up to that, I just wonder whether anyone can look to see whether or not any of the chaperones might bind small stuff. So I give you the end point, which chaperone is actually the last one on where it's or actually bring small stuff to learn.
So So Lila Pierre from David Stern's lab, the paper which he published in 2015 on RAP1, no, RAP2, sorry, Rubisco accumulation factor 2. She did some very, how do I say, crude cross-linking studies to try and identify any binding partners with RAP2. And they proposed in the model, based on some very tenuous pictures in the supplementary data, and David agrees with me as well, so that the small subunit in the RAP1 and RAP2 may actually interact. Okay, but again, no one else has been able to reproduce that, and the work which the Harpers have done in E.coli suggests that association may not occur. But we don't know. But it could actually be RAP2 in small subunit, which is what I hypothesised about 12 months ago. And that's one thing which we're testing using our new system. So, we'll see.
More general question. So how much does rising CO2 in the atmosphere affect photosynthetic efficiency of everything? Temperature in water is the same. The rise doesn't make much difference.
So rising CO2 on its own affects different plants differently. So we saw rice, it improves photosynthesis, improves growth. That's not the same for tobacco. You hit a ceiling at a very early rate. So in other words, if you grew a plant at 400 ppm and then 700 ppm, the plant at 700 ppm will actually be smaller than 400 ppm. We've done that experiment.
Why is that?
It's because we're talking about the push. So we're talking about photosynthesis. It's making product, but it needs to go somewhere, and that's where you've got this. What is the sink, the pool? Is there anywhere for it to go? If there is, then you're going to see this continual improvement. If there isn't, then there's this feedback saying we don't need this. So therefore, what they do is they literally knock down Rubisco amount. So it's not a universal response. That's my working hypothesis at the moment. Carbon allocation is actually really critical in actually, if we're going to improve photosynthesis, so what? If we don't actually then start to play around with, where does that additional carbon, where's it allocated to, or can we play around with that? Then yeah, it's going to be a waste of time. But if you join the two together, CO2 and heat, well, these sort of experiments are pretty rare to be done. Most people just focus on one or the other. They don't usually join them together. But there are some base experiments, so free air CO2 enrichment experiments done in Illinois and also in Sydney now, where they're looking into that sort of thing. I'm not actually overly familiar with that literature, but it's pretty new. It's a bit of a trade-off. So there is no simple answer, essentially. So yeah, and you'll see some face experiments. Some plants show continued improvements in photosynthetic capacity. Others don't. Others actually reduce it after a couple of generations as well. So things like soybean, there's improvement in the first generation, and after that, productivity actually declines back to essentially ambient CO2 levels. So it's complicated.
That's because subsequent generations make less control.
Well, they don't ever measure those things properly. So yeah, what they do is they base it on a gas exchange and they parameterize and work out a VC max and make an assumption about, well, this plant has this much Rubisco, as opposed to actually going in and measuring. Because there's all sorts of other parameters which can affect that measure. So, give it to me, I found. Which is what I was doing in Bob's work for a long time now with Rubisco.
What would Rubisco has too many limitations, and the solution is coming from other completely different enzymes.
Oh yeah. So they've done many years ago. They did a flux analysis on all the other enzymes in the carbon cycle and identified acetylheptulose 1,7 phosphate, SPPase, as an enzyme which is limiting the regeneration phase of Calvin cycle. And what they did is they knocked it down a little bit using the old antisense. This is back in 1999. And yeah, they saw this a very small reduction actually had a dramatic effect on the growth of the plant photosynthesis. So then they did the reverse and overexpressed it, and they saw improvement in photosynthetic properties. And so this has been going on now for a long time. So they've now done this in wheat in the UK, and the field trial started last year. In the glasshouse, they saw 20% improvement in growth and yield of these wheat plants, overexpressing SPPAs in the glasshouse. Problem is that their glasshouses are actually high CO2 glasshouse, because there's no glasshouse which actually is ambient unless you scrub it. And so it sort of makes sense to us that that would be the case at 25 degrees. But SPPAs is actually a very thermo unstable enzyme as well. So, and we haven't heard anything about their field trials of their wheat. So we don't know whether or not they saw success or they saw nothing. And again, this is one of the targets which was engineered using tobacco. Again, they did field trials of this with the right program, and they saw improvements in growth. But I think, yeah, you always got to be worried about these growth experiments. If you don't actually look at the young plants, you may actually find that all they've done is they've just got early bigger, and that the actual growth rate in the field is exactly the same, but because they're early bigger, you're already 2 days ahead. And 2 days ahead for tobacco plants through an exponential growth is around about 20% combined max, about 12 to 15 centimetres combined. So we'll always step forward that. Do you want an addendum to that? The addendum is that last year there was a paper that came out in plant cells which shows that 2-phosphoglycolate inhibits SPPA. So therefore, you can actually get the same phenotype by actually increasing 2-phosphoglycolate phosphatase. By reducing 2-phosphoglycolate levels, the SPPA will actually fully active